So hi all and welcome to another Spring Knowledge Transfer event. Thank you all for joining us today, along with Northumbrian Water, Northumbria University and Newcastle University, who will talk us through their journey so far with the Ofwat funded SuPR Loofah project. This involves the design and creation of an innovative loofah for removal and recovery of phosphorus from wastewater, which prevents it from causing damaging algal blooms which can suffocate local ecosystems. So for those of you who aren't already familiar with Spring, we are the Centre of Excellence for Innovation in the UK and Ireland, and our main objective is to enable effective delivery of innovation and collaboration by creating centralised intelligence for both water companies and suppliers. Our mission is to attract, connect and support innovators across the industry to accelerate transformation. And that's why we facilitate and host knowledge transfers events such as today. Through knowledge sharing, Spring supports the transfer of learnings from an innovation project to the wider sector, creating project legacy that helps innovators to identify and adopt the right solutions at a greater pace. Last year, we partnered with Ofwat, the economic regulator for England and Wales, to build a view of what best practice knowledge transfer actually looks like. We brought together senior leaders and innovation managers to design a vision for knowledge sharing. What actually is it that we're trying to achieve? And ultimately, the answer was that we want to unlock faster adoption by creating a world-class knowledge sharing culture. So the 6 principles we created were: number 1, learn from what already exists before committing to innovation projects. To aid this, we created the Spring platform, which hosts a multitude of project case studies aligned to the priorities laid out in the Water Industry Strategy 2050. Links to both of those resources will be put in the chat in a moment. We are also working on other tools to ensure innovation intelligence is easily accessible to the sector. Number 2 is target innovation insights to the right audience to meet our objectives. So we want to make sure the right people are actually in the room listening to these lessons that we're sharing. So for today, do we have our scientists, innovators, algal experts on the call? And do we have the right decision makers from other organisations who can take this knowledge back to their own organisation? The 3rd principle is insights are regularly shared through multiple channels from day 1 of the project. So historically, we know knowledge shares happened at the end of a project, and we want to start moving towards a model where we share from day 1, and it's more than just a final report. Number 4 is that sector knowledge is open and centralised. So we really want to make sure that we're all being open and honest when it comes to the knowledge from innovation projects, and we want to make sure it's all in one place, i.e. Spring, because fragmented knowledge is a lot more difficult to engage with and learn from. United Utilities do a great job of this, and I'll give an example in a moment of where we've seen further sector success come out of their knowledge sharing efforts. Number 5 is that insights from both innovation successes and failures are shared. As we know, the lessons learned from failures are just as important as the lessons of success, and we'll hear about some of those from our presenters today. Number 6, and the final one, is that people are empowered to receive and embed lessons from external sources. So Spring and presenters on the call can all share knowledge, but we want to make sure that the receivers of the knowledge actually have the resources and capacity required to make that change, and that they feel empowered to do so. Again, I'll go into more detail in a second, but Northumbrian Water are a great example of where teams have been empowered to take project insights and learn and even expand on them. So coming to that great example, a great time where these principles have come into play is with the Water Industry for Infrastructure project. This was one that was showcased by Spring last year alongside United Utilities, Changemaker 3D and Manchester Metropolitan University, who came together to share valuable insights from the Water Industry for Infrastructure project, which was funded by Ofwat and led by United Utilities. As part of the showcase, they talked through the implementation of the project trials and the promising results seen so far, such as 25% lower carbon and 20% cost savings through the use of 3D printing. These insights are now driving a sector-first implementation at Northumbrian Water, another industry leader in innovation, who have progressed in plans for the UK's first underground water asset created using mobile 3D printing. Thanks to United Utilities' open sharing of innovation insights and a real desire to raise the quality of knowledge sharing in the sector, Northumbrian Water have been able to easily learn from the project and expand on it, rather than having to start from scratch. From this, we can see that knowledge sharing really does lead to more experimentation, faster paced adoption of solutions, and sooner benefit realisation. By driving knowledge sharing efforts across the sector, United Utilities are also gaining a reputation as industry leaders in innovation and collaboration, bolstering their reputation amongst customers and regulators alike. So I just want to say a quick thank you to both United Utilities and Northumbrian Water for engaging with Spring to drive knowledge sharing and innovation across the sector. Back to today, I just want to give a warm welcome to all the audience members who are joining us live. We are joined by the UK and Ireland water companies, supply chain, academia, and lots of overseas colleagues too. So welcome to the call and thank you so much for joining us. We'll be linking a poll at the end of the chat and at the beginning of the break, as well as the end of this showcase, to find out how engaging you found today, and also if you are hoping to use the information shared today and in what format. So please do submit your answers when this comes through. It really helps us shape the kind of content that we have in these showcases. You'll also get the chance to ask the project team any questions at the end of the session, so make sure to keep putting your queries in the chat throughout today's presentation. So on to today, I'm just going to run through the agenda and introduce our lovely speakers before handing over to Richard to kick the session off. So we'll start with an introduction from Richard Warneford, Wastewater Director, covering Northumbrian Water's perspective of knowledge sharing and the value that it can bring to individual companies and the sector as a whole. We'll then get some more context behind the project and go into an overview of the technology from the academic partners. We'll hear from the project manager about benefits seen so far before going into a short break. After that, we'll do a deeper dive into the learnings and challenges of the project and finish up with our usual Q&A session. Speakers-wise, we'll be hearing from Richard Warneford, Wastewater Director, Rhiann White, WINEP Strategy Manager, and Alex Corner, Project Manager Technician, all from Northumbrian Water, as well as Justin Perry from Northumbria University and Phil Chen at Newcastle University. So with that, that is me done, and I'll hand over to you, Richard.
Okay, hi everybody. So as Leah said, my name is Richard Warneford. I'm Wastewater Director with Northumbrian Water. I also chair an organisation called UK Water Industry Research, so UCWIA. So been involved with Spring, which was kind of created alongside UCWIA really from the very beginning, and really passionate about Spring and what it can do for the sector. So I wanted to say a few minutes this morning, just kicking off this session on that perspective. I'm going to start by, and there's a link here, I'm going to start by talking about something we do in Northumbrian, which is the Innovation Festival. So the Innovation Festival, I think we're into our 9th year this year. The theme last year was all about doing things at pace and scale, so speed and scale. And at the session last time, we had 3,200 attendees from 650 organisations. We had about 37 universities involved, and people were there from about 39 countries. So the reason I mention that is what that's allowed us to do, and the whole sector to do actually, is to think very differently. The more different thoughts you get in a room, the more different partners you work with, the more you're going to get better ideas. And lots of you hopefully have been to the previous festivals. I'd like to extend the invite to all of you in your organisations for this year. 7th of July is the week. But the things we've created from a session like that is fantastic. So moving towards Spring, because there's a similar theme here for me about knowledge sharing. The more we kind of collaborate and the more we think about how we do things together as a sector, the better. We're kind of compared and contrasted in the water sector. There are all kinds of different league tables. But sometimes your advantage is quite short-lived, in my opinion. If you're higher or lower in the league table, and I think there's a lot better things that we can do in regaining trust and confidence in the sector by working together. And I think what we do through Spring, and I think we should all be sharing all of our innovations, all of our knowledge sharing through Spring, whether it's Ofwat funded, and I'll say a little bit about that in a second, or otherwise. I think really we should get behind this and use this vehicle to make sure that we share better. You've had a great example from Leah there around the 3D printing. So I'd just like to echo that one. That's the one where, if you like, other companies were leading, we've listened to that knowledge share. And what I would say give us was confidence, kind of validation of what we were already doing ourselves, and some ideas, and that kind of confidence to go on and crack on. So I think that is a really helpful thing. So moving on to SuPR Loofah and what you're going to hear about today, I'm certainly not going to do any of the technical stuff today on it. but sustainable phosphate recovery. For a number of reasons that you can all imagine, this is a really exciting project, something we're really keen on. We're a bit sad on the wastewater director. We really love algae, so sometimes it can cause us the odd problem on the water side of the business on impounding reservoirs and things. But for us, it's really exciting. We've got the green machine down at our Brand Sands site, and then you've got the super Loofah you're going to hear about today. And from an environmental impact point of view, fantastic, really great opportunity there as well. But I guess also beyond that, to recover something which is, I guess, a scarce product and do things in a more circular economy type way is certainly the way we should all be heading. So from my point of view, stick with spring. Really make sure that you're using it as a good knowledge share opportunity. Hopefully you'll enjoy the session today. And I guess my last message on any session like today is you kind of get out of it what you put into it. So please think as you go through. It's useful to the speakers to get questions and get a bit of discussion going. So think about that as you go through, and the more interactive we make it and the more we get from you, the better. So have a good day. Thank you.
Morning, everyone. My name's Brianne White. I'm part of the strategic planning team at Northumbrian Water, and I've been involved in the Super Loofah project from the beginning. I should just say this is a project that's been run through the Water Innovation Fund. So Northumbrian Water came together with Newcastle University and Northumbria University to put together a bid which we were successful on, and that's led to this collaboration and this project going forwards. The slides. So what's the problem? I think probably a lot of you from the water industry are already aware of the problem of phosphorus, and that removing and recovering it is a problem for us in the water sector, and we need to do that to protect the environment. Phosphorus leads to eutrophication, and that, of course, we know is a problem in our watercourses, rivers and lakes. Looking at protecting the environment, we're quite heavily regulated on our phosphorus in our discharges to the environment. And we know that by 2027, 95% of sewage treatment works will have a P removal permit on it to stop any additional phosphorus going to those watercourses. I've been involved in something called the Chemical Investigation Programme for quite a long time, and that project looked at different phosphorus removal technologies back in 2014 to about 2020, I think. And we covered quite a few different technologies, and we trialled them to look at how low you could remove phosphorus with traditional technologies. Of those technologies, only 5 didn't use chemical addition. However, we believe this Super Loofah is going to be the way forward. We don't need any chemical addition. We're able to remove phosphorus using an algae-based technology and then recover that phosphorus, which could then be part of the circular economy in terms of a fertiliser. So I'm going to quickly talk you through our outcomes and plan. So we're talking about 2 different disciplines here, which is microalgae and growing, and that's where Newcastle University come in with their expertise on the algae. And then we're looking at a coating so to make a biocomposite layer that we can fix those algae in position. And that's Northumbria's expertise. So you'll meet both those guys in a minute, and they'll talk you through the kind of chemistry and the biology of the project. But what we intend to do is prove the use of these biocomposites for P removal in wastewater. What we want to do is go from that proof of concept, technology readiness level, kind of at the 3 zone, and move right through to number 5 on technology readiness level, which is pilot scale. And we've got the pilot scale rig up and running now at Birtley Sewage Treatment Works in the northeast, and that will allow us to then do some further scale up and look at where the commercialisation will sit on that. So I've got a little diagram on there where you can see we go through phase 1 right through to phase 6. As Leah said, we were talking about knowledge sharing, so we're here at kind of phase 4, doing it fairly early on that, and that all links back in with that knowledge share piece that we've been talking about earlier. So just a quick one on the team, and then I'm going to hand over to the universities to talk you through the science. So we've got a big team, but a good team here. So we're centered around our program manager, Alex, who you'll meet at the end. and we've got a delivery team comprising us, Northumbrian Water, Newcastle University, Northumbria University, and then we're also supported by Welsh Water. We have some reporting to do via the Off-Water Innovation Fund, and then we've got a couple of people that sit in terms of process engineers to help us with the technical side. At Newcastle University, we've got Dr. John Lee and Dr. Carrie Caldwell, and then Phil, who you'll meet in a minute, to talk about the biology. And at Northumbria University, we've got Professor Justin Perry and Dr. Matt Unthank, and then Craig Hodgson, who's done the work with the biocomposites. So they'll talk you through all of that. And I'm going to hand over to Justin now to start with chemistry. Chemistry is at the root of everything. Over to you, Justin.
Thank you. Hello, everyone. Yes, so I'm Justin Perry, and yeah, so if I start off with this slide here, I'll just say that I'm representing a whole team of people who have been doing this work. So we call ourselves SPIN. We're the polymer and materials group at Northumbria University. And on this first slide, I'm just going to sort of talk about the top line of that circular diagram. So you want to remove phosphorus. You need a material to do that, and we were really focused on what sort of material we could actually use to do that. So if you look in the green box to the right there, you can see that there's sort of different components to make up a material. You need the coating, you need the algae, the thing that looks like sort of a long brown objects, that's the loofah, that's the sort of support for it. You put those three things together and you get the material to the right hand side, and that's the material that you're going to use to remove the phosphorus from the water. So if we look at a little bit closer then, what's the background for this? Well, the background for this is a bit of work that the same team from Northumbria and Newcastle University have been doing for a while now, which is looking at trapping microalgae and similar microorganisms within a layer and seeing if they can interact with their environment. So we initially done this looking at trying to remove carbon dioxide from air, and this is a nice piece of published work, which was the first piece of thing we've done together. So a bit of chemistry coming now. So across the top there, there's some diagram, a diagram which is a nice chemistry diagram, which people may not be completely aware of how to interpret, but it's very simple in that you can see that there are three components into what's the coating, which is where the algae live, and that goes to make a plastic or polymer. And if you do that, you've got to make sure that your organism is very happy to sit there and live. And you have those different components in there. They interact in a different way to make sure that it's a robust treatment, it's porous, and it doesn't swell and break. So we worked on that with the group. We worked out how to get these things to sit in water for a long while, absorb carbon dioxide, and that seemed to be quite nice. But obviously you can try other things that these arrangements can actually work. So this biocomposite we felt had other options. And in fact, it's quite general. We call it a living biocomposite. The technology for the plastic coating that sits over the loofah, it's very similar to house paint. If you've got a bathroom and you've got grime living in the top of your bathroom, that's effectively doing the same thing under the paint that we are trying to do here, but we're doing it in a very much more useful sort of way. So you just mix in the organism that you require in with the paint. The paint dries, it slowly dries away and leaves a layer that is porous, but yet a nice place for these microorganisms to thrive. They're able to take things in from their environment, and they can live for a long while. So we've had 70-day lengths of time where these things have lived. We literally haven't looked longer because that's how long a research project lasts in a lot of cases. So our experience is making these sort of polymers. So we've done it at this is a 400 mil scale, making it. You can see that one of the reasons we get into chemistry because we like building nice rigs, and we've built apparatus to make it at this sort of small scale. We've got a background where we've made lots of different sort of polymers and paints. So we've got all the materials to make it into a nice smooth formulation. So the coating that goes on the loofah is just like a paint. You can dip it, you can spray it, and that's what we've done. So in terms of this, what have we done? Well, what we want to do is we want to be able to trap the organisms on top of the scaffold underneath the coating so that they can live there, interact with the environment, pull the phosphorus out. So we knew we could do that. Phil is going to tell you through about all the biological results. We did other things to make sure we knew what was happening. So this is an example here. We built a version of our polymer which was fluorescent, which is quite nice. The loofah itself is fluorescent. The algal cells have a little bit of inherent fluorescence, and the polymer, we made a new polymer which was also fluorescent as well. So in these two diagrams you can see, and Phil has got another one as well, so I won't steal his thunder. We can actually look at how effective our material is at keeping living cells on top of the scaffold where it's happy to live and do its job. So we're able to work out how these things were working and look at it in a sort of using all the techniques of biology and chemistry. Okay, so that's okay. Now, one of the things we were quite keen to do was, going back to that nice cycle, you can see that there's as well as looking at across the top, you've got to realize that once we've made the thing, and it's sat and absorbed phosphorus, what do we want to do with it as well? We wanted that to be a recyclable material. Loofah is a biological material, so that's going to work okay. The next thing is, well, the coating. What can we do with the coating? Well, the coating, the polystyrene element in the coating is there for toughness, but it's a petrochemical based material. So we were trying to move away from that, and this is something we've been working on within our group for a while. What can we do to make polymers more biosourceable, more biodegradable? And one way of doing that is swapping out the petrochemical components. So what we did as part of this project, and another project we were working on simultaneously, was create polymer alternatives which work exactly the same way as this coating material, but have no obligate petrochemicals in there as well. So on the right hand side you can see there's more of my lovely chemical diagrams, and what we've done is we've completely replaced the polystyrene element of it with materials which are made from starch or cellulose or chitosan, which are all natural biopolymers which are effectively waste materials or have waste roots. And we've done that, and it works just nicely. So you can see there's a gloved hand sitting there holding a piece of plastic. It literally is like a normal piece of plastic. It has the same polymer properties, but it doesn't have things which have to come from petrochemicals. We've replaced most of the all the polystyrene with petrochemical, with this material, and the other materials within there are ones which can be made from biosources as well. So that works really well, and that's quite nice, and we're currently patenting that for obvious reasons, because it looks like a good idea. So what happened after that? Well, what we've done is we've scaled this material up. We've now got so there's Craig, who's been the very capable pair of hands in the laboratory doing all this work. We can scale it up, and we've made 40-litre batches of this, which is what you need for the pilot plant that's currently operating here in Berkeley. It works very well. It's biosourced as a material, the coating, and it's able to perform all the things you need for the microalgae to sit in there and operate, taking the phosphorus in and incorporating it. So it's working really very well. So one thing that we did, which is sort of the next step, we're looking ahead what we can do better in future on this, and one of the aspects of it was the loofah. The loofah is fantastic for the basic material, which is the scaffold for all this. You want something which is available with a high surface area. So that's a really great thing. So loofah is a vegetable which you can grow, but we don't grow it in the UK. So we were looking for other things that we could do, and other things we could do we could use for this. Well, another project I was looking through British industry to see what sort of materials are that have high surface area and could possibly be used. So British wool is a material which has similar properties. So one of the things we did during this project is we've looked at wool. You can scour the surface of the wool, and the coating material attaches to it, just like it did with that loofah. You can and you can see on the right hand side there is an example of a large piece of wool where we've actually put this coating on as well. So that's for the future. The current material we are doing is the current material that we're using is loofah based, but we are currently looking at other things as well. So that wool is an example of that. So that's the end of the chemistry. I will hand over to Phil to talk about the biology.
Hi everyone, I'm Phil from Newcastle University. So I'm partially based in chemical engineering and partially based in marine biology. So the marine biology bit is more about growing the algae, and the chemical engineering is more about scaling up everything. So Justin has explained the concept of biocomposite. So what I'm going to talk about is how to use this biocomposite to remove phosphate. And first of all, the algae we use is Chlorella vulgaris. So it's the most commonly cultured algae, microalgae. And it's also reasonably kind of desiccation tolerant because our coating process involves desiccation. So we need to make sure they can survive the process and they can be revived once they're being rehydrated. So once we mixed everything together, we put algae onto the loofah. So the first thing we need to know if they have like a securely attached to the loofah. So here's some microscopy images. So on the left is the confocal image. So all the red dots are individual algae, and the blue thing is the loofah, and the green is the latex that Northumbria developed, and they bind the algae to the loofah surface. You can see it's quite evenly distributed. And on the right is the SEM image, where you can see all those in like a kind of spherical object, kind of embedded in a layer of coating. So that's all individual spherical objects are individual chlorella cells. So by these conforming microscope images, we can confirm that we have quite uniform coating with algae coated on the surface of the loofah. So it's our biocomposite. And yeah, then we need to test if this biocomposite can be used to do phosphate removal. So to do this, we designed a system which we would call a lab scale phosphate removal test. So what we have is a customly made photobioreactor, which is the one on the top left corner, so basically they're made of perspex and they're clear and they're about 10 ml in volume. So we can insert a sheet of loofah biocomposite into it. And then we have on the left of the image is a reservoir bottle with 1 liter of wastewater. And we can pump the wastewater through the biocomposite and then go back to the reservoir bottle. And we pump continuously non-stop for a few weeks, and we take samples in between and analyze the phosphate concentration in the liquid sample. And we also do media change in between to give them fresh phosphate. So we have 4 reactors running in parallel, and they're testing 4 different latex formulations. So we have the starch latex, and we also have the petroleum-based latex. So when I compare the performance of different latex, and also we add some nanoclay into the formulation. Nanoclays are kind of a filler that can increase the porosity and the permeation of the polymer coating. And this is a typical like a test result of the phosphate removal test. So what we can see here, all these red arrows is where when we do the like a media change, which we use like 5 milligram per liter artificial wastewater. It's diluted from the BG11 culture media. And the time is not to the scale, just to show broad idea of like what it look like. So in the first week, from day 0 to day 7, we hardly see any phosphate removal. It is because the algae need time to recover from the desiccation, and they also need time to kind of get used to the new environment in the bioreactor. And from day 7, after the media change, you can instantly see a drop in the phosphate level. And however, at this point, you won't see like an entire elimination of the phosphate. It didn't go to 0 like instantly. But over time, if you look at the next 3 water change, they immediately go to 0 in the first few hours of the liquid change. So it means the phosphate kind of removal rate is kind of accelerating. And this is what being like observing for most of our experiment. And we didn't we run the test for a bit longer, but we didn't show the results there because after like 2-3 weeks of test, we see the results getting kind of varied. and more unpredictable. But what we can see is like within 3 weeks, all the results are very predictable and all the phosphates being removed after the water change. So this is the comparison of the phosphate removal rate of like four different like latex formulation. So what you can see here is like in the beginning, certain formulation do perform better. In this case is at day 7, if you look at it, it is the petroleum-based latex that's performed the best at removing phosphate. However, what we can see here is that over time, all other groups also kind of catch up. And if you like look at day 11, so basically all like a biocomposites with different formulation, they all be able to kind of remove phosphate, like a kind of most of the phosphate within the first hour of the liquid change, and remove almost all the phosphate after 4 hours. And yeah, basically this kind of saying that the formulation do matter, but it's the effect is kind of limited, and the starch latex that the Northumbria University developed is good enough to have like to be used as a kind of biocomposite for phosphate removal, and they're kind of the more other advantage. So they're kind of better than the petroleum-based one. And there's also one thing worth mentioning is that because the way we designed the experiment, so these phosphate removal rate is kind of slightly higher than if it's a continuous like a flow of like a constant phosphate level. This is because we have kind of starvation period introduced after the like full removal of the phosphate and the next water change. So basically the starvation will cause the phosphate, the algae to be more active after the phosphate is kind of replenished. But we think it's desirable for our kind of experiment because we can see a more distinct drop in the phosphate level, and also it appears much like nicer on the graph. So we also previously it was the synthetic wastewater, but we also experiment them with the real wastewater. So we took the real final effluent from here, Berkeley, and we measured it's about 2 to 2.5 milligram per liter. We conducted 2 experiments with 2 different batches of water sample. And we have this kind of normalized data showing we also see a very similar pattern to what we saw with the synthetic one. So basically we see a drop that's the day 7 to begin with. And then at beginning it was quite slow, but it kind of catch up with the reactivity. So this is the visual appearance after 3 weeks. So basically we can see for all 4 samples, they're kind of brightly green, which means the algae are kind of quite healthy in them. And also worth noting that because none of the experiment is kind of in a sterile condition, so we have other things growing in them, and which I'll explain later. But the overall idea is they can stay healthy for about a week, about a month, and they can keep removing phosphate. And so we took some sample after finished experiment, we took some biocomposite sample out, we dried them and look at them under the SEM. So there is some evidence of like a biofilm formation on the surface of the biocomposite. So basically what you see here, all those kind of still like spherical shape things, they are kind of just based on their shape and size, we can tell that they are chlorella. So they're forming on the surface of the biocomposite, and they're forming these kind of EPS extracellular stuff, around them. So our guess is that some of the algae, they kind of embedded in the coating, they kind of overgrow the space inside the coating, and they kind of migrate onto the surface and spill their colony of biofilm on the surface. And also sometimes we have some other stuff growing in the biocomposite with algae, and we haven't kind of identified like all the species, but we have kept some sample, and we have sent them for DNA sequencing. So we will have more data in the future to tell us what they are. But right now the most important thing is during our experiment, we didn't see any negative influence of these kind of other things growing in the biocomposite. And actually on the opposite, I mean, actually we sometimes see an increase in the kind of phosphate removal with those like other thing when there's other things growing with the algae. So the next step, we proved kind of the phosphate removal works in the lab, and now we are going to scaling everything up for a pilot plant kind of scale test at Berkeley. So to scale up, it's quite dramatically from 10 mil, we like a upscale up to like a 95-liter water tank, and we adopt it into a bioreactor. So we just find the largest Loofah we can find in the internet, and we dip the whole Loofah into a mixture of algae and latex, and then we dry them, and then we kind of cut them and layer them into the reactor and insert light tubes into it. So this is the ongoing pilot plant trials. So basically we have the equipment, the rig ready, and we have kind of built them right next to the final effluent well in Berkeley. So we draw like a real effluent from the well and feed them into our biocomposite. And you can see there are light, like LED light there. So they'll be inserted into the biocomposite and provide the photosynthesis. So because of some delay in the kind of like in the application of this device, so I mean, because we're not really experienced with the wastewater, and there was some issue with like building the rig, but everything is ready right now, and we'll start our like a test like in a few weeks here at Berkeley. So if we got some new results and there's other means of like sharing it, we will kind of keep updating our like progress here. Okay, that's it for my part. I will hand it to Alex.
Hello, I'm Alex Corner. I'm a project manager for Northumbrian Water. I predominantly work on the wastewater networks. So this is a bit of a different project for me. It's been really interesting to kind of work with the universities and the whole project team on this. So I'm going to talk about the next steps. So as Phil's just mentioned there, we've got the field test up and running now. So we'll be starting the testing soon, and that should run for a couple of months up into June. Going forward, we'll be analysing the data and moving into phase 5 and phase 6, as Rianne was saying, evaluation and reporting, and then disseminating the results. We will also be able to do a cost benefit analysis, so we'll have some information on return on investment for what that'll look like if we scale this up. So that analysis will also go into the scoping of scaling up if we go into a full pee removal on an actual wastewater treatment site, and use it as the whole pee removal for that site, or if we come up with something that you can add in the summer to increase when you have more pee removal needs. We're also looking at the options for total nutrient removal testing, so that's phosphorus as well as total nitrogen removal. Well, we're producing a case study which should be coming out within a few weeks. So that will outline the findings from the project, as well as getting some of the results from the cost beneficial analysis will be added once we have that. It's going to be updated as we get results, so keep your eyes out for that. So the benefits: the solution could provide phosphorus removal lower than traditional methods such as ferric. At the moment, that brings it down to 0.2, which is the technically achievable limit. We've seen that reduced to as low as 0. We've also got a proven removal rate of 2 to 3 milligrams per litre per hour. Another benefit is that we're going to be removing the need for chemicals if you use this solution. So there's no ferric, and then also when you use ferric, you need pH correction quite often. So we don't need that either, which will help contribute to reduce it to our net zero emissions goal. if you don't need to have the chemicals delivered to site as often at all for this. We're also looking at potentially creating a circular economy. So as we're looking at recovering the phosphorus, we could potentially use that as a product rather than a waste product, and sell that back to farmers. As Justin was saying, that we're looking at using the starch rather than the plastic, so that and we've done testing to see if it is biodegradable, which it is, which would allow it to be used as a fertiliser. So going further forward, we're looking at further optimising the system that we've put in place here, and potentially looking at other things such as wool, as Justin was saying, and then other options that might come out. We're also looking at potentially going beyond phosphorus, as I mentioned, nitrogen and other metals. And I know there's some forever chemicals that we could look at potentially being able to remove as we go forward. Looking at potentially a multi-algae in seaweed treatment, so you would have different types of algae in a seaweed, which would, so you can remove different pollutants or chemicals with those. And we've also looked at an idea of a rapid response mobile treatment unit. So that's something that we could quickly go out to where we've seen algal blooms and protect the environment, as they're quite dangerous to the environment there. That's me. Yeah.
Perfect. Thank you, Alex. And thank you to all our presenters so far for a great presentation. We are just going to go to a quick 15-minute break, give you a chance to get some coffees, go to the toilet. You don't need to leave the call or anything. All of your cameras and mics are off, so please stay on the call. You just come back in 15 minutes time. While we are on the break, I just wanted to point out an upcoming showcase we have for water efficiency. If anybody is interested in today's showcase, any other kind of similar topics, or water efficiency as a topic, do go ahead and sign up. You just scan the barcode using your camera on your phone, and it'll take you to the sign up page and more information. Along the bottom there, you can see the kind of themes that we tend to cater these knowledge sharing showcases towards. My colleague will also put a link to the poll that I mentioned in my introduction in the chat in a moment. Anybody who's planning on dropping off in the break, please just give us an answer on the poll before you go. It really, really helps us make sure that these are the most valuable they can be for our audience. So with that being said, we're going to go to the break. I'm going to round up to give you a little extra time. So we'll come back at 11 o'clock on the dot to carry on with some lessons learned and the Q&A. Please do keep putting your questions in the chat through the break as well as they come to you, and we'll address them at the end. Thanks.
You two white-shirted people.
Hi, everyone. Welcome back, everyone, from the break. Thank you for rejoining us. As I say, we're now just going to go into hearing a deeper dive of some lessons from all of the presenters together, and then we'll be going into our Q&A. So do keep asking any questions that pop into your mind. And with that, I'll hand back over.
Okay, great. Shall I lead this little bit, guys, as soon as I did the slide? We were asked to have a little think about lessons learned from the project, things that went really well and things that didn't go so well. So we've had a kind of ongoing process with that as we've done, because we've done some reporting for as well from the Innovation Fund there to kind of really enable that kind of sharing piece to go from the things that didn't work as well as the things that did, because that's important both ways. So I'll probably start with the things that were not so great, because it's always good, and then we'll end with the good stuff. So we had a bit of learning to do there on our kind of academic versus industry commitments. Obviously, it's been a while since I've been in academia, but yeah, there's some holidays and students' commitments and teaching commitments to go on. But also from us in the water industry, we had some awkward things like business plans and final determination and that kind of thing on the go. So there was a few little bits there that we had to kind of work around each other's commitments. So having advanced knowledge of that would be kind of useful going forwards, I guess, to allow everyone to kind of understand those time commitments and how they worked for different people within the different organisations. We had some very normal delays with rig construction and third-party kind of bits where they were delivering stuff and it was a bit late. And some of that we then led to a little delay because we had to get the power supply sorted on site, and we couldn't do that before the rig was delivered because we weren't sure exactly what they would need, and there were a few little bits with that, and one delay led to another delay. Fairly normal stuff, but still something worth mentioning. The bigger the project, the more parts you have, I suppose, the more risk from on a delay point. We had a little issue with seasonality. Algae obviously like it warm. Burtley in January is not so warm. So we had a kind of heater unit to ensure that final effluent temperature was brought up a little bit to encourage that algae growth. I think if we'd started it in May or June, it wouldn't have been a problem, but the heater had to do a kind of fairly manful job in the 6th of January when it was -4 here. So yeah, so we had a little bit of issues there, and we had to upgrade our heating unit at that point. And then obviously you've heard a little bit about our wool substrate. That one was a really good thing to investigate, but maybe wasn't for this particular trial. But it's not to say that we don't want to look at other substrates and find things that are waste products that could be used in this. We're again still thinking of other things that might fit that criteria, whether it's a UK grown loofah or some other kind of high cellulose content substrate that would work from that. So that's our kind of list. Anybody else got anything to add to that while I'm here? No? I'll keep talking. Then moving on to things that are really good. So the team's been really consistent all the way through. So we've had the same teams from the universities and the same postgrads. So Phil and his colleague Craig at Northumbria. So that's been all the way through a really good team. We've had really regular, consistent meetings. And I've put that under that comms section and the biscuits bit because we didn't do them all just over Teams. We're all relatively local, but we all have different commitments, and it would have been really easy just to resort to a monthly meeting on Teams. But actually, some of the progress we've made and having that sharing and kind of ideas around a table with a cup of tea and a biscuit while on site looking at the rig has actually been really, really useful to take forwards. And it might sound very basic, but actually it was a really useful bit there to kind of move us forward. And then the final bit I've put on there is about site visits. We appreciate that us in the water industry, sewage treatment works all the time. It's not that exciting or interesting, and there's aspects of that we might forget to explain. But if we go out with these guys and we show them and we look at where would be a good place to put a rig and everything, we get some really good interactions, and it really feeds back through the project so that there's really good understanding on both sides of what the kind of aim is and what we're trying to achieve and how we're going to make it work in real life as such. So for me, that's where we got to with those lessons. And anybody else wants to comment on good things?
Yeah, well, I'll say as one of the academics, it's always really good if you're working on a sort of a commercially oriented thing to get out to the university and actually find out what the situation is. So yeah, Northumbrian Water were really good at showing us what the issue was and showing us the plant and how it would work. And if you're thinking about scaling up, and in our laboratory, we don't ever do anything other than over about sort of 5 litres in one batch. And coming to a place like this to work out what the scale is really gives you an idea on how you're going to have to scale things up. So that was a really good part of the project.
Okay, I think are we moving to questions? Yeah, you're running the show. So.
Thank you everybody for sharing your learning so honestly and openly. So that does bring us to the end of the presentation, and we'll now go into some questions and answers. I am just going to stop sharing the screen so you can see our lovely presenters a little bit better. Just what you want to hear. Yeah. Perfect. Okay, perfect. So let me just make sure everyone can see us. Perfect. Okay. So first question for Justin was from Sarah, who wanted to ask, is the coating used biodegradable?
Now there's a good question. So when we started off, the coating we used, the one that was polystyrene base, has some ability to biodegrade in the same way that sort of paint does, but it's not great. So that was one of the things that we were working through in all our projects, is trying to work out how to make things more sustainable and biodegradable. So as part of that, we were looking to change the makeup of it. So that's why we've completely removed the polystyrene coating and replaced it with the polysaccharide. So the version, Phil's used the phrase starch. We are using non-food grade starch as the major structural component of this. And we, as part of what we're doing with this and other projects, we're setting up a lab resource to monitor the biodegradability using the existing ISO standards, just to see how they're going. We've started. It's certainly better at biodegrading than your standard acrylate wall paint, which is good to know. We can see that from the methodology. We haven't finished. We haven't finished the tests to know exactly how good it is. Anybody who's a chemist out there will know that there will be issues with some elements of it. If you've got an acrylate chain, that's something that's fairly commonly seen in plastics, which we have to deal with. We have ideas on how to remove that, and that will be the next iteration of making our materials more biodegradable. But currently, we can see it. In fact, that was one of the issues when we were doing it. Myself and Matt, who's the other program lead on this, we were thinking, why are we making a paint out of a potential food for a microalgae? Aren't they just going to chop it all up? But that didn't seem to happen. So yes, we believe it's biodegradable. We reckon we can make it better, and that's the next stage.
Great, cool. So something that's going to be explored further in the...
It is being explored, yes.
Oh, fab. Amazing, thank you. So another one for you, Justin, while you're in the spotlight, is from Nadia, who wants to ask, what is the required surface area of the support?
Right, so we actually approached this from the other way around. What we did was we went and found a sort of a material scaffold which had the highest surface area. So you can look around and see a whole range of different things that could possibly be it. The Newcastle team had lots of experience using Loofah, and that was the carbon dioxide one, was how we used that, and that was chosen on the basis we knew it had a massive surface area, and we knew that we could paint it or coat it using the materials, so that's how we did it. We don't have a number for how much surface area it has, because literally we are just picking things which have acknowledged high surface areas, and that's how we got to wool. That was the next thing.
Perfect, thank you. Did you try anything else other than wool in the testing?
No. We were focused on Loofah because we knew we had to do it for the plant here, and at the same time, as part of a project where we were looking at different materials across the UK which were relatively cheap, and somebody from Wales, or Welsh University, was saying, do you realise how available wool is? And as long as you can scare it to make it acceptable for coating, it's available. And that's why we moved on to that.
Oh, cool. Okay, great. Thank you. So one for you, Rhiann, from Tim is, may I ask if the SuPR Loofah project has considered the implications of contaminants and of emerging concern, such as PFAS solving into the microalgae? This could affect the use of microalgae as a fertiliser on agricultural land. And as a potential solution, could the effluent pass through a granular activated carbon filter before phosphorus recovery on the Loofah? Long questions, if you want to repeat any of that.
Yeah, thanks, Tim. Yes, we have. We've been thinking about what the aspects are. We know algae can be quite keen on uptaking other things like metals or heavy metals and other forever chemicals, those PFASs and PFOSs. And what we would look at is, obviously, if we were going to utilise the algae, if they turned out to be super good at that, then that would be one way of removing those from a waste stream. But then obviously it wouldn't be viable to go back on the land. But what we could be looking at is specific algae that would remove specific contaminants and have them in series. Or, as you say, put something upstream where we're making sure we're removing those contaminants first to enhance that kind of circular piece and making sure that what went back to the land was just your N and P. But we're in the early stages of that yet, but they are risks that we're kind of very aware of, and we've got them kind of written down and noted, and they'll be part of our kind of case study and further thoughts going forwards.
Perfect, thank you. And again, while you're in the spotlight, one from Matthew is, at what point in the works do you envisage this being used, e.g. at a secondary or tertiary stage?
Yeah, so I think for me, at the minute we've got it on as a tertiary polishing unit, and that seems to be where it would sit naturally, taking those P levels from, you know, I'm thinking about the 3s and 4s down to the limits of detection, really, where we're getting to with that, and what we've shown, and that would seem a natural bit. I suspect there'll be applications where we're looking at some really small works, maybe descriptive permits rather than numeric, and that will allow that to kind of be an add-on at the back end. We might have other applications where we might put it on a side stream treatment. We've done some work with algae for side stream liquor treatment at some of our other works, different setup, but still maybe applicable. So there'd be some kind of commonality there where we could look at putting it on the site.
Great, thank you. Justin, back to you. So a question from Matt for you is, what concentrations of synthetic copolymer constituents are in the influent and effluent?
Okay, so the coating, the plastic coating that goes on the loofah is literally painted on the loofah and it's dried, and then that whole unit is put into the flow cell. There is, you know, the water going in is flowing over a dried plastic, plastic coating. So influent, no. One of the things that we were worried about was, you know, how quickly it would break down and you know, effectively see even macroscopic changes like flaking away. We haven't seen that. At the molecular level, it's quite possible things might break off because, you know, we're running water over a surface. We're currently getting the tests together to monitor that, and, you know, sort of at the molecular level, and microscopy to check surfaces to see what happens when you get a crack and a break. And you know, we've shown you the techniques you can do to look at that sort of thing, which is quite nice. At the moment, I would say that you know, we're not seeing a huge breakdown over the surface lifetime of the plastic, which is good, but we will be monitoring that. We're seeing what it goes. In terms of, is it a microplastic? Well, it's a fully formed plastic piece of plastic. It's, you know, it's a thermoset coating with links across between the particles. That's how you can see it coalesces. So it, you know, it's not going to be releasing microplastics, you know, sort of without some sort of breakdown process. But that breakdown process is something we're looking at, and something we're trying our future and our current and future research is making sure to minimise that, because we realise that's an issue.
I'll just jump in there because I sit on our microplastics group, along, I suspect, with some of the people in the audience. So I've got that on a key message that we can't be adding any microplastics.
Yeah, absolutely. I understand that.
Perfect. Thank you both. So Phil, coming over to you, Sarah wanted to ask, what kind of maintenance is anticipated for this system?
So, I mean, regarding light and heating, it's all kind of automatic, so there's no like labor needed. And for the only bit I can think of that involve labor is when we need to change the biocomposite. So basically, it really depends on kind of what lifespan our biocomposite has. What we can confirm is it has a minimum of like 1 month of lifespan. So after 1 month, we can see some of the loofah structure kind of deteriorating, become kind of less kind of disintegrated a little bit. So if that happened, then we kind of, or say if we see it's kind of saturated with the phosphate and there's no longer any phosphate removed, then probably we need to kind of replace the biocomposite. And yeah, that's the only kind of maintenance I would think of.
Great, thank you. Justin, back to you again. You're a popular one.
Yeah.
From Matt, is the synthetic copolymer polystyrene, or the replacement latex component a microplastic?
Okay, so first of all, going forward, and not just because we've invented it, I would suggest we don't pick the plastics for the polystyrene in it. The current view from the tests are that as Phil has said, that, you know, there is no functional advantage to the polystyrene component. So I would suggest moving to the ones which have the highest biosourcing. Now, as I said to you before, I mean, there are elements of you know, polymer chemistry in there, and we are looking to remove that, but they are not unusually long pieces of plastic. So yes, I understand that, and you know, as Rihanna said, there's something we have to look at, but you know, that's the next step, I would suggest.
Perfect, thank you. And just to add on to that, how much of the copolymer substrate is lost during treatment?
Okay, right. So how much is lost? You know, I've sort of answered that one before. We haven't seen gross changes to the plastic coating on a polymer before. It seems to work pretty well. We are currently looking at it in a sort of molecular level. I have a master's student, a very talented lad, who's working out a mass spectrometry method to track chemical loss from these sort of plastics, and we will be doing microscopy analysis just to work out, you know, what sort of cracks and how it works, that sort of stuff. So yes, that is something we are looking at, but you know, these things are not breaking apart in the lab over the months that they're running, which is good.
Great, thank you. Phil, back to you. Another one from Matt. What does the removal rate mean in terms of amount or area of asset needed?
So I think removal rate is not really our a major concern, because what we know is that comparing to suspension, I mean biocomposite certainly has advantage of kind of intensifying the process, and certainly need much less footprint for the same type of things can be kind of that need to kind of remove phosphate. I mean, especially if you have a pond or something. So what we know is our system is kind of have high concentration of algae in it, and they have a much, they should have a much faster removal rate comparing to a suspension system. So, but I think our advantage is more in the kind of removing those phosphate with quite high, low level of phosphate, because we know that our system has a kind of capacity, it will saturate eventually. So it's not really designed to remove a huge amount, from a really high phosphate level. So it's kind of removing trace amount of phosphate, and then kind of allow the biocomposite to gradually kind of saturated with the phosphate, and it can be kind of recycled for fertilizer use.
Yeah, I think I'd probably add to that while we've got the pilot rig up and running now, we'll be able to get some really much better numbers on that. One of the bits we'll be looking at is what that flow rate is from we're obviously taking out of a final effluent chamber, so we'll be able to change that rate that we're passing it across. And once we've got our baseline data, we'll be able to do some optimization there and see which bit which is the optimum flow rate for the loofahs and the removal rates, and we'll be able to really update on that and get some volumes and concentrations removed, which I suspect my colleagues in the water industry they'll want those kind of numbers as well. So it'll be nice to be able to share that.
Great, thank you, Rianne. And yeah, as you say, we can add those updates to the case study. So anybody who wants to stay up to date as the trials continue, yeah, do log into the platform and have a look at the case study. And I'm sure we'll let you know on LinkedIn every time that's updated. Great. Okay, so coming to you, Alex, from Matt, who wanted to ask, have you run the process as continuous process rather than a batch one?
So at the moment we haven't. It's just been batch ones in the lab, but we've got the pilot rig all set up now, and we will be starting the continuous process, I think, within the next couple of weeks.
Great, exciting times. Sounds like we'll need another showcase, actually. Cool. So going back to you, Phil, how does performance vary with different temperatures and intensities and daily durations of sunlight?
So the performance definitely will vary, but in our case, because we want a more controlled environment, so basically we controlled everything. So we have artificial kind of LED lighting. So it's set at 16 hours light and 8 hours darkness. And for the temperature, set at 20. So it's not really going to vary much, because we have a heater installed to bring the water temperature to 20. So yeah, in the real scenario, if we don't implement the heating yet, mean that the reactivity would be higher in the summer and probably no reactivity in winter if it's just room temperature.
Makes sense, thank you. Hey, so Alex, Rebecca wanted to ask, what happens to loofah and algae at the end of its kind of use life, I guess?
Yeah, so ideally this is where we bring it into the circular economy. So once it's got the phosphorus, we would be looking at how we can go forward and potentially use that as fertiliser, or like rather than it being a waste product, we can use it as a product for other industries potentially.
Great, thank you. No, circular economy is a big thing in the water industry at the moment, so great to see this contributing to that. Phil, coming back to you again from Rebecca, does it need to run under UV light, or can natural light be used?
Natural light can be used. Yeah.
Just don't get much of it in England.
It's not under UV light. It's sunlight is what is the wavelength that's being used.
Okay, cool. But it can be used under natural light when you have it.
Yeah.
Great, cool. And then one from Chris is, what are the potential benefits over an algal bioreactor, which may not need the support structure?
The support structure basically intensify the kind of the amount of algae in a unit volume.
Okay.
So basically kind of encouraging them to form biofilm instead of form like suspension. And there are some research suggesting that whilst many of the algae, they kind of in the nature they're kind of in suspension, but most of them can be kind of encouraged to be kind of converted into biofilm state. And they also suggest that there's like more kind of biomass production or something like when it is in this state.
Great, thank you. Justin, coming back to you, does the mobile treatment unit mean you think it could be used in raw water to remove pee?
Either.
Right. Okay. So right. So I'll answer that one. So this is one of our myself and Matt's sort of desires. We'd really like to try this on as a sort of mobile treatment unit. It's just in our experience of sort of chemistry, and in chemistry today everything's moving to sort of a bespoke operation and sort of continuous flow over batch. So we think there's an opportunity there to do this. And just going back to the polymers for a second, for the coating, is the coating that we've got there has lots of opportunity to be made for a bespoke thing. So if there is an issue that people there is a worry that there would be release or breakdown of a plastic component or the algae into the water, then we can change the robustness of that plastic. We can make it stiffer and harder. We have to make sure we don't make it so that there's nothing able to transfer between across the coating barrier into the algae or something. So I think there's plenty of opportunities to play with the system we've got for different opportunities, and that one where we're looking at different ways of doing things in different sources of phosphorus is something we want to look at.
Great, thank you, Anne. Do you want to add anything to that?
No, I just think that probably feeds nicely, and I think there is an aspect of where we would be looking at from a clean water side, there would be maybe some aspects of that alongside your mobile bit. Wherever we're storing water, there's obviously a risk sometimes, and that could be kind of seasonally applicable.
Great, thank you. Alex, coming back to you, Ewan wanted to ask, is there a ballpark size of water treatment works in which super loofah would be feasible? For example, less than 250 population equivalent. And is the algae population susceptible to certain types of loading, say a high source apportionment of trade effluent? Are there any pollutants that will destroy the algae?
So I'll answer the first part.
Yeah.
But I think somebody else will be better for the second part of that question. So I would imagine for if we were to use it for the full treatment, it would probably be the smaller scale treatment works, and then potentially, as Rhiannon was saying, in the summer when there's more phosphorus, and we could probably use it as an addition on some larger treatment maybe. I don't know if anybody wants to add to that or the second question.
Yeah, I think probably I'm seeing it as a tertiary unit on a smaller site, and then there'll be some applications where we might want to, if we do get it mobile, move it around. I'd like to see things such as addition of solar panels or wind power to generate some of that lighting element, and if we were going to do it in the winter, then to make that kind of energy requirement reduction, if there was wind or solar, I guess would be some kind of renewable there would be an option. In terms of the, what did you ask? Oh, the loading and the pollutants. We haven't seen that as yet. Chlorella a fairly robust algae, but I'll refer to Phil on that if we think anything kills chlorella. Does it? Any chemicals? Yeah. But so far so good. Bertley hasn't caused it any problems yet, and it's got a fair amount of trade effluent going into Bertley sewage treatment works. So normal, majority domestic, but with an element of trade. So it's a good test base to do it on.
I would say it's just like algae, they're tougher than we would imagine. They can survive many tough situations.
I think the other thing to point out, of course, is that it's not floating around in the water. So that in the same way the phosphorus has to go across the polymer membrane, the pollutants, the effluent would have to do the same thing. So that might slow it down. So, it could be that you might be able to divert it away if something suddenly came in that you didn't want in there, because, you're not, there's a bit of a time before it will go across the membrane.
Okay, thank you everyone. And is there potential in a country like England to store up that kind of energy over the summer and then make use of it over the winter when we don't have the sun?
I would imagine so, yes.
Yeah.
I'm not an expert on battery storage, but I do know it is something that.
Sorry, wrong person to ask.
I think it's just because I was opposite you, wasn't it? But yeah, I'm sure that will come in terms of that storage element.
Perfect, thank you. We're into new territory now, so I'm not sure who to aim these at, so feel free to jump in if you know the answer. But the next one is from William, who wants to ask any more information on the trial site at all, PE, flow rates, and if the catchment has any traders.
Yeah, so I can answer that. So Bertley's a fairly average size works just outside Gateshead. I haven't got the PE directly to hand. It's a trickling filter site, but we're taking a kind of element of the final effluent through the unit. So is it 4 litres a minute, Phil? I can't remember what the flow rate is. We're taking
here.
Yeah,
4 litres.
Yeah, 4 litres a minute we're going to take out for the pilot rig. I can supply anyone with any additional data they want, specifically on Bertley. If anyone wants to come or send an email, that's fine. The catchment has a little bit of trade, but majority domestic with some trade effluent.
I guess that'll all be included in the case study as well.
Yeah, well,
share out.
Perfect, thanks both. Okay, next question is how is the algae population controlled, / how is excess algae removed from the system?
So we don't have a control system right now. The idea is the algae we're using is not genetically modified, so it's safe. Even they kind of released into the final effluent, and there should be, if there is a problem, then we should add a kind of like filter system to it to kind of stop it. And in the lab, it's possible to kind of coat multiple layers to kind of prevent the algae from kind of escaping the biocomposite, but basically it's more, can be realized in the lab, but it's more difficult if it's a large scale production. What's the other part? Yes, yeah, I think that's it.
Yeah.
Perfect, thank you.
Just one other thing is, as has been explained to me, I am not a biologist, is that the algae that sit within the polymer coating are not reproducing, and they are metabolizing, but they're not spending their time at the same rate as a natural population expanding. So they are turning over the phosphorus and taking in, but at the rate we can see, but that we're not seeing massive population growth, which would be a problem with them bursting through the surface, for instance.
Great, thank you. Question from Paul, who wants to ask, I assume you need to provide aeration. How is this done? from outside or through the loofah?
Oh, it's my question. So in the lab, we aerate the reservoir bottle, and on site, because we think the final effluent, they kind of run into the well, so it's kind of, and we pump it directly into the reactor. So there should be aeration when the kind of the water transporting.
We've got a DO probe on that as it's going through the unit, so we can monitor any particular drop in dissolved oxygen, and we have kind of considered that as an option if we have to aerate, but we're not anticipating we have to at the minute because we're looking at quite high dissolved oxygens going in, and that flow rate's enough hopefully to get it not seeing significant loss through the process.
Cool, thank you. Okay, so next question from Gillian is, does extreme temperatures impact the performance of the algae at all, and where does the process sit within the treatment process? I guess it's kind of two separate questions.
Yeah, I guess the second part we have answered a few times, haven't we? So we know it's going to probably be tertiary.
Yeah.
As we mentioned in our lessons learned, we did have some elements. I think the algae like it warmer. And part of the reasoning for us to heat that effluent up was to stop any time delay of developing that and having to wait with the project. The algae grow perfectly well in normal final effluent temperatures. We just picked 20 because that's what they'd used in the lab. Certainly one of the trials will, once we've got this baseline, is looking at temperature, and we won't be heating the effluent up here over the summer because we'll be letting it do its thing naturally. So we're not anticipating any problems with that, but we'll have to see what happens come October. Time.
Great, thank you. I apologise now if I have any repeated questions, but it's hard to Great. Question from Ewan is, what numeric consents is the system suitable for? Is it just targeting phosphorus consents? And is there any data for the minimum lighting requirements?
Can't do lighting, but I can do the P permitting. So at the minute, the Off-Water Innovation Fund was specifically to look at P because that was part of our scope. We haven't gone beyond that. Not to say we wouldn't like to, and we certainly would, but that will be the kind of next stage. So we're certainly looking at P at the minute, and we can prove that we can go down way below the current TAL of 0.25. I think you've got to 0.002, did you, Phil, down on the algae removal? So that gives us a remit. We're putting between 3 and 4 milligram a litre in. So we're looking at quite good removal rates there. I think that is probably fairly typical of final effluent. We did struggle a little bit with finding a site where we didn't have a P permit to put this on, so we weren't removing totally down. But yeah, that's where we're at the minute, and we'll be happy to supply some more data once we've got those kind of baselines for that. The pilot rig up and running all of April and May, it should be. So we should have some really good data coming out of that very soon.
Perfect, thank you. Is that including the lighting?
Oh, the lighting bit, yes. I don't know how much lighting you have to apply. Lighting's not mine.
I can't remember it clearly, but the benefit of having a kind of biocomposite system is that the light kind of decreased, the light intensity decreased quite dramatically over distance. So we always, in what we did in the lab is always provide maximum that we can. And so basically, even if the algae at the surface is not the ideal for them, the one kind of buried a little bit beneath it will be the kind of perfect condition for the next layer. So basically, we see like about 90% of the light kind of lost after 2 centimeters of the loofah. So they can't really penetrate very deep. So that's why we kind of always use the maximum light, like if that we can.
And it's 360 degrees around the loofah, isn't it?
Yeah.
Great, thank you. So next question from Gillian. I think we already had that one actually. That's a repeat. That is about size of treatment works, which I think we've already done, haven't we? Rena has asked, do you see this technology being able to treat high phosphorus concentration in crude sewage to final effluent requirements once it is scaled up? Or is it more of a secondary P removal used to trim down already low P concentration?
I think at the moment we're looking at tertiary units and that kind of medium to low level. So I think, as I say, we have used other algae units to look at higher concentrations. So I don't think it's impossible. I think it's just not within the scope where we are with this project at the minute, but it would definitely be one to look at later. Certainly when you've got getting the higher concentrations, you need to be a little bit careful about making sure you've got the right algae and that it's acclimatized to that. So within the scope of this, it was for, you know, final effluents and a polishing type unit, but not to say that we couldn't go on and do it. And Northumbrian Water are very keen to use algae, so we have been looking at it for other things. I think you've put some links on to the algae project we've got. So.
Can I add a few words? Because basically the algae we use chlorella, and I guess most of the algae, they don't have a kind of reservoir for the phosphates. Not like carbon, they were built like a lipid just to store it for future use. They don't store phosphate. So I mean, so that's why I mean it would be they will get saturated. So if it's a very high phosphate level, it means it will be saturated much faster, and then we need to replace the whole biocomposite. So, but if there is someone that knows something about kind of algae that can do kind of phosphate storage, then there won't be any problem. We can just leave them there, and they can take as many phosphate as they can.
Perfect. Thank you so much. And Alex, you kind of covered next steps of the project. Is this something that you'd be looking into in the next steps at all, or is it kind of more way in the future once we've actually scaled this up? And
I guess it's something that we can look at. I don't think we've looked at it just yet, but I know we were talking about potential on the water side, and then this could be another thing that we look to see if it's something that we can do.
Great, thank you. Next question from Ewan. He's asking the temperature of the heater. What's the temperature that the heater had to bring the water to? Our design guidance says a design at a minimum temperature of 80 C. Is this consistent with algae treatment?
Oh yes, I think the 8 applies to nitrification, if I'm right on what Ewan's thinking there. And the phosphorus removal is slightly different. We aimed for 20, which that was why we had a problem with the heater in Berkeley in January, because fire level was pretty chilly. But it's been proved in the lab, I think, to work from between 8 and 16 is the kind of norm, and then up at 20 is where you want to get to for that really quick acclimatization and utilization. I don't know what the upper max is, what the algae would survive at, but I presume pretty warm.
Great, thank you. Next question, can the algae be grown on an inorganic substrate like redundant biofilter media?
I think the answer is yes, but also the question brings back to the surface area. So what we find is Loofah has a really high surface area. So if we can have similar system but inorganic, I mean, we can try to coat it onto it. We can give that a test.
See, and just as someone who's not an expert myself, what do you mean when you're talking about the surface area of the Loofah material?
So basically, the more porous, the more like it is, it's more likely it has a higher surface area. Okay. So if like a ceramic or like a stone biofilter media is like a solid, it basically can only grow on the surface. If it's porous, it can also grow in the inside.
Okay, great, thank you. Okay, cool. So next question is from Paul, who wants to ask, does the process algae also absorb CO2?
Yeah, yes.
Great, that was a nice quick one. Cool. Coming back to a question from Matthew, what is needed to convert the algae into a fertiliser once it has been removed from the plant?
Good question. I think that's part of our next steps. I'm imagining at this point it's a drying process and then a grinding process to get it into a form that is bindable, and then that you can spread it, and we look at how quickly that dissolves on the land and it doesn't wash out, and that's that side, and how much bioavailability there is across there back on, which I think indications so far from the decomposition tests we've done seem really good. I don't know kind of how much volume you get from a ground down loofah in terms of thinking about farmers and big bags of fertiliser going to fields, but that's all a very answerable question, so I'm not worried about that side of it.
Thank you. Next question from Paul, who wants to know, does the substrate need to be fixed in place, or could it be suspended like the media in MOB?
Good question. For those of you that aren't aware, MOB stands for mobile organic biomass. So that's a system where you have a fixed biofilm but on a suspended substrate in a treatment work. So it's kind of the crossover between your traditional trickling filter versus an activated sludge. So it's the current kind of thinking. I don't see why not. It's again just comes down to that surface area and then what aeration you would require. And if we are talking algae and a lighting aspect, obviously that there's some element of that going forwards that we'd have to think about. Yeah, good question.
Great, thank you. Next one is, how successfully have you managed to extract phosphate and capture it for resale so far?
You done that in the lab yet?
We have.
You have, yeah.
So actually, I prepared slides somehow disappeared. So basically, we dried and we grind and we digest the biocomposite after the phosphate removal test, and we can see a quite sharp increase in the phosphate content in the biocomposite. So basically, that's if you want it to be kind of in a kind of liquid phase, you digest them. If you just want like a powder phase, then you just grind them.
Great, thank you. Cool. Next question from Jeton, who wants to know, how often would the super loofah need to be replaced in the unit? Has it been considered being used in conjunction with nature-based solutions like wetlands?
So we've tested it. So far, we've tested it for up to a month, haven't we? And so potentially longer than that, but we haven't got the data for in the trial just yet for how long it'll last. But at least a month.
Again, want to kind of update the case study as you go.
Yeah, and the wetlands bit, yes, I definitely foresee that, you know, this could be an element that you add to your nature-based solution. I don't have a final vision of what it would look like, but it definitely would add to that, especially if you added that solar power or renewable resource from if there was a lighting requirement.
Great, thank you. Coming up to the final question, so if anybody else does have any kind of final thoughts or anything they want to add in, make sure to pop it in the chat now so I don't miss them. Okay, so next question is from Paul again, who's asking, how long does each loofah element last and how often do the elements need replacing? Again, that's a repeat of the... Great, okay. And then how much power does the system use for lighting and heating is the final one.
I think right now it's a 15 kilowatt power supply, right? So everything is drawn from a 15.
I think it was 32. I think it was slightly over 16.
It's 16.
So there was 16 or 32. I think we had to use a 32.
So slightly over.
So this is something that will be in the case study. So we've started looking into this so we can advise to work on the return on investment.
Perfect. Thank you all. That is the final question by the looks of it in the chat, which is great. We do have a couple of minutes left, so again, if anybody thinks of any other questions, do put them in the chat. I'm just checking if we've had any more in privately at all. I think we've answered all the questions. I guess the only other one to ask is this the first time that Northumbrian Water have kind of experimented with using algae for wastewater treatment?
No, we do have a really good relationship with Newcastle University and Northumbria. We've done some work with algae earlier with the team there, and we have our own algal treatment unit at our big site called Bransands on Teesside. That is at the moment, it's called the Green Machine. You can go and look it up. That was on the news a couple of weeks ago, so there's some good visuals there. It definitely looks very lurid green. It's very nice to see. That is one where we're looking at treating return liquors, so really high strength effluent for phosphorus removal. So that's a slightly different way of looking at algae treatment, but yeah, certainly something Northumbrian Water are really keen on, and I feel we're kind of utilising quite a lot of local knowledge, and it's really part of that going forwards, and should have a really good impact for customers and the environment, because we're trying to explore all the best ways to use the algae, and that can only be a good thing going forward.
Yeah, definitely. Great, thank you very much. Justin, you might have already touched on this one, but just to make sure I've got everything answered, how biodegradable is the biocomposite?
Right. Okay. So I would suggest we are in our design brief for this is to make it as biodegradable as possible. So all through the project, we've been looking at it and saying, is there a way we can make this as biodegradable? You know, and that's the reason why we swapped out the petrochemical elements, because they're not going to be so biodegradable, and move to the other one, move to sort of starch or polysaccharide based components. As I think I said to you before, that I don't think it's the perfect answer we've got at the moment, but I think we've gone a long way to prove biodegradability using biosource components, which is usually a good start. So I think we've got a first iteration which is strong in those terms. You've got to think that the coating is not the major element in terms of the mass of this, that actually that the major element is currently Loofah, which is obviously very biodegradable, and that's why it was chosen. So yes, the component that is not, you know, as biodegradable as we can make it at the moment is a minor component, and we're working on making it even less of an issue.
Great, thank you. And again, this might have already been touched on, but just to make sure, I know I had a couple of people messaging me about this before the showcase. So just to kind of wrap up and summarise, in the next kind of phases of this project, is there potential that this technology could be used for clean water treatment as well as wastewater?
Alex, do you want to take that one, or do you want me to?
I'll let you take it.
Yeah, I think there is. Obviously, I'm not a clean water specialist, but I don't see any reason why not. We certainly know we have problems with algae sometimes when large volumes of water are being stored, especially seasonally. So that kind of aspect of it would definitely be applicable. I can't speak now. And that element of, could you just utilise something when needed, be it mobile or be it a turn on? I think the bonus of having the algae embedded in the loofah is that there's a very little growth time requirement. It's almost ready to go straight away. They're ready to uptake it. You're not waiting on something growing for a few weeks. While you build up a population, they're there and ready to go, which is something that maybe we haven't touched on, but actually is probably quite an important feature of this, because it's almost instantaneous when they get it, rather than that growth period.
The statutory stuff's working a bit better than the mobile.
Yeah, I think so. On that, and the point about embedding, having that surface area, you can get more algae on one per unit volume of whatever we end up using loofah at this stage, and that will allow that speed of uptake to kind of really impact. And when we're trying to respond to environmental issues, then speed is of the essence. So that's a really good point to make.
And in terms of the coating, we can change the depth and the robustness of the coating to suit the application. That's just a formulation problem. That's not new science, really.
Great, thank you. And I guess just one more final question for me. I know adopting solutions at pace is a big thing that's kind of in talk at the moment in the water industry. Is there any kind of tips and tricks from Northumbrian Water's perspective that could help somebody adopt this kind of project at a greater pace at all?
I think from me, I would say is get your team around you and get it brought in and try and work to that end point. We've been working to that end point of getting something on site right from day one. That was our aim, to get here and get it working. Things, you know, different elements and phases of it move at different speeds, but as long as you're all working to that end point together, it tends to move things on. Once you can show people as well and bring them, that kind of creates its own kind of enthusiasm, which stimulates that project more and opens that up to other commercial kind of opportunities.
Great, thank you so much. We do have a couple more questions actually. How best can we keep up to date with the results of the trial? So I think that would be what you said, isn't it? So we'll be updating the case study.
We'll be updating the case study as we go on, as we get the results. But there's also Ofwat page that we'll have a link to, I'm sure, which has some information on there as well.
Great, Thank you. Yeah, and again, for anybody who didn't get it in the intro, this event will be recorded and posted alongside a written version of the case study on our website, which is what they've been talking about when they've been talking about updating the case study. So if you head to the Spring platform, which the link was put in at the start of the session today, and I'm sure it'll be put in again in my outro. If you head to that platform and just create a membership, if you're not already a member, it's entirely free. It takes 30 seconds. And yeah, you'll be able to see all of the stuff that we've talked about today in there, along with the kind of slides that we used and as I say, the recording from today. So do head over there, and we'll be keeping that up to date as we learn more from the next steps of the SuPR Loofah trial. Cool. So I think that brings us to the end of the session. We haven't had any more questions. Let me just triple check. Nope, perfect. Yeah, so that is the end of today's session. Thank you so much again for everyone who took the time out of their day to come. Thank you again to our fantastic presenters who have shared the learning so openly and honestly, and have really made today an engaging, informative piece. Again, thank you everyone, and we'll see you soon. Bye.